Hello class, I am Mrs. Gabriella Herring, and I brought a few scientists over to help us talk about transcription and translation. To start us off for transcription versus translation, I wanted to mention the central dogma. The central dogma is the flow of genetic information to make up functional proteins. In the central dogma, it goes from DNA to RNA to proteins, and in between those steps is transcription and translation. In transcription, it goes from DNA to RNA, and in translation, it goes from RNA to proteins.
Hi, my name is Jalen, and I'm mainly going to be talking about the differences between transcription and translation. To begin with, transcription is found in the nucleus of eukaryotic cells and in the cytoplasm of prokaryotic cells. The process is the messenger RNA copies the DNA as a blueprint to the ribosomes to later start translation. This produces a non-coding RNA, such as tRNA, mRNA, rRNA. This starts when the RNA polymerase protein binds to the promoter in DNA. Then the promoter directs it to the exact location to start transcription. While translation is found in the cytoplasm in prokaryotic cells, since they do not have membrane organelles, and in eukaryotic ribosomes on the ER. Specifically, the ribosomes decoded by the ribosomes to produce the amino acids with the help from tRNA. The products are just mRNA to proteins, and it begins when tRNA binds to the starting codon message from the mRNA.
For transcription in prokaryotes, it's a less number compared to eukaryotes. In prokaryotes, it takes place in the cytoplasm. The enzyme involved in transcription is the Hall enzyme, which consists of RNA polymerase and the sigma factor. There are three stages to the transcription in prokaryotes, which is the initiation, elongation, and the termination. In the initiation, the sigma factor helps lead the RNA polymerase to the promoter regions. For the RNA polymerase to know which strand to bind to, there are specific sequences in both the promoter and terminator region for the RNA polymerase to know where to bind. Once the RNA polymerase binds, the sigma factor is released as it's done with its job, and the stage of elongation begins. That's when RNA polymerase begins transcribing the gene to make the product of RNA. For prokaryotes, there is only the coded regions. There are no uncoded regions known as the introns. This then leads into the termination stage, where RNA polymerase falls off as it no longer needs to code because an RNA strand is now produced, and the RNA polymerase can continue transcribing by the sigma factor rebinding to it and starting the process all over again. Now, compared to eukaryotes, prokaryotes do not have mRNA processing, which is the addition of a 5-cut, RNA splicing, and a poly A tail.
In eukaryotes, transcription is a little bit different from that of prokaryotes. Transcription can be split into 4 phases: initiation, elongation, termination, and processing. Each of these steps is vital to making an RNA molecule to be translated. In order for initiation to occur, instead of using the sigma factor to determine the location of what gene to transcribe, eukaryotes have many primers which they can use. Once a primer with a specific nucleotide sequence that matches with the DNA sequence of the gene is trying to replicate is located, it will attach to that specific part of the genome and will label that strand of DNA as the template DNA. Once this occurs, an RNA polymerase II will recognize that the primer is present and will start to unzip the DNA by breaking the hydrogen bonds between matching base pairs. The polymerase will then bind to the primer and start transcribing a new strand of mRNA, which ends the initiation phase. After initiation, transcription goes into its next phase, which is called elongation. Elongation begins when the polymerase slides along the DNA template strand and grabs free nucleotides that float around the nucleus to match them with bases of the template strand from the 3 to 5 end of the template DNA. The big difference between replicating DNA and making RNA is that RNA polymerase attaches uracil instead of thymine to the template DNA. RNA polymerase is very quick when it is in the elongation phase, as it attaches around 40 to 80 nucleotides per second, in which there is a 0.00001% chance that an error can occur. When this happens, it can cause a massive change to the nucleotide sequence of the mRNA that could cause alternate splicing to occur or other issues with translation. At some point, the polymerase will see a specific set of nucleotides, which will end the elongation phase and begins the termination phase. Termination phase begins when mRNA polymerase reaches the termination site. When the polymerase hits one of these sequences, it will stop, release the pre-mRNA, and then unbind for the template strand. After termination, the pre-mRNA must become a mature mRNA. For this to happen, the pre-mRNA goes through the processing phase. It begins with an enzyme called a spliceosome, which splices away introns or uncoding regions from the mRNA, only leaving exons or coding regions. Once the spliceosome unbinds, the mRNA gets a modified guanine nucleotide placed on its 5 end, which is called the 5 cap. This cap helps protect the mRNA from unnecessary degradation. On the 3 end, a long string of 8-9 nucleotides is attached, which is called a poly A tail. The poly A tail makes the mRNA more stable as well as protects it from degradation. Once these processes are finished, the pre-mRNA is now a fully mature mRNA and can go to the ribosome to begin translation.
Hi, I'm Alyssa, and I'm going to talk about translation in prokaryotes and eukaryotes. In prokaryotes, this process requires special coded regions along the mRNA molecule called the Shine-Delgarno sequence. Once this region is found by the small ribosomal U subunit, it assembles the large subunit and uses the closest AUG site to begin translation. It then moves on to elongation, which is a progression of the ribosome along the mRNA. This reads the codons and arranges amino acids due to the genetic code. After it then moves on to termination, when amino acid is terminated into a polypeptide chain. The ribosome is then disassociated, and the mRNA and polypeptides are released. And then in eukaryotes, the initiated tRNA binds to the small ribosomal subunit. Here, the 5' cap on the mRNA is bound by the initiated tRNA complex, which scans the mRNA until it reaches the AUG site. The initiated tRNA then settles on the AUG codon, and then the large ribosomal subunit is recruited along with other initiated proteins. Then, just like in prokaryotes, it moves on to the elongation phase. Since the first AUG codon is always used, though, the same polypeptide is encoded.
Hello, my name is Jennifer, and I'll be going over errors that occur in transcription and translation. Mistakes during transcription and translation will occur no matter what. On average, a cell makes thousands of mRNAs during transcription and will make about 1 error per 10,000 to 100,000 bases. Cytosine becoming uracil and substituting the base is one of the most common types of transcription errors. This mainly occurs due to the deamination of cytosine into uracil, and it often causes mutagenic uracil guanine mispairs or uracil adenine pairs. Improper base pair placement is also another issue that occurs when mRNAs produced incorrectly. And some diseases that come out of transcription errors include cancer, autoimmune disease, neurological disorders, diabetes, cardiovascular disease, and obesity. During translation, the average process of translation causes 1 error per 1,000 to about 10,000 codons translated. Aside from errors that carry on from transcription, translation errors can include improper codon coding. These errors are counted as nonsense or missense errors. Nonsense errors are when ribosomes prematurely end the translation of a coding sequence, while a missense error is when the improper amino acid is incorporated into a peptide chain. These translational errors, or more generally misfolded proteins, can cause diseases such as Alzheimer's, Parkinson's, Huntington's, cystic fibrosis, and other degenerative disorders. Of course, these harmful errors don't occur often enough to cause an overabundance of faulty proteins. There are also ways to fix such possible errors. One example is that polymerase II can backtrack to correct errors found in faulty mRNA in order to match it with the template DNA. This only occurs in eukaryotic cells, of course, as prokaryotic cells don't contain polymerase II. Another example is that the preset coding for amino acids has a safety net. Since it's possible for different codons to code for the same amino acid, the alteration of a base might not impact the end amino acid result. This will allow the protein to correctly form despite having an error. Finally, if the protein ends up misfolded anyways, then they'll go down the proteasome route and can be commissioned.
Hi guys, my name is Jose. How are we doing with similarities and differences in transcription between eukaryotes and prokaryotes? Starting off with similarities between eukaryotes and prokaryotes in the process of transcription is that both use a DNA to create a template called mRNA, and as a result, RNA or mRNA is a result of both processes in both cells. So RNA polymerase is the facilitator of transcription in both eukaryotes and prokaryotes. Now going on to some differences in transcription between eukaryotes and prokaryotes, we go over that transcription occurs in the cytoplasm for prokaryotes because prokaryotes have no membrane-bound organelles, and thus in eukaryotes, which do have membrane organelles, the transcription process occurs in the nucleus, which is the housing of the genetic material of the cell. Thus, the DNA is copied into the membrane. So some other differences is that the RNA molecule is capped with polyA tail, and is capped and a polyA tail is attached to the end in eukaryotes, whereas in prokaryotes it does not happen. In eukaryotes it does occur because it has the mRNA has to be stable enough to leave the nucleus and make it to the ribosome. And another difference is that signal factors exist in prokaryotes, whereas GTF factors exist in eukaryotes. Signal factors are what attach to the DNA, and GTF is also attached to DNA, but they are different in both prokaryotes and eukaryotes. Then going on to the next slide, we have some similarities and differences between translation in eukaryotes and prokaryotes. Starting off some similarities, mRNA is used as a template for both processes. tRNA, as a result, is used in both to bring amino acids into the ribosome, and amino acid chains are a result of both processes. Another thing that is similar to eukaryotes and prokaryotes in the process of translation is that ribosomes are the tool or machinery used in the production of the amino acid chain. And tRNA, which does bring in the aminos, ties it together the amino acids, and thus its peptide bond is created to amino acids, and the amino acid chain is created. Some differences between eukaryotes and prokaryotes in the process of translation is that translation occurs immediately after mRNA has been transcribed in prokaryotes because, of course, they have no organelles, and so the cytoplasm is where these both processes occurs. Whereas in eukaryotes, translation is a bit slower because the mRNA has to leave the nucleus and then go to the ribosome, which will take a bit longer. And thus, because of that, mRNA molecules in prokaryotes have a short lifespan, whereas in eukaryotes, they have a longer lifespan. And that wraps up the differences and similarities in transcription and translation, and also the process in which DNA goes to protein. Thank you.
All right, thank you to our scientists for coming here today to talk about transcription and translation. Thank you.